of the gene at post-transcription, post-transcription and post-translation level. As I said that at transcription levels, immense amount of study has immense amount of studies has already been conducted. A thorough understanding of gene expression level at post-translation modification, post-translation, post-transcription will helpful in the development of new strategies that will enhance the plant stress tolerance. So, to understand the transcription, so the rationale of this study is to understand the complete repertoire of microRNA and miRNA along with the targets. So this information is not available. The information about the presence of microRNA in garden pea plant is not available in the literature. The information about the presence of miRNA in biotrophic pathogen is not at all available in our literature. So the goal of this study is to fill the gap in the collected literature by providing a detailed analysis of microRNA in pea plant, miRNA in fungal pathogen ECP and miRNA involved in interaction between ECP. So the finding aim to enhance our understanding of miRNA mediated regulated network, ultimately contributing to improve garden pea plant. So the thing is, when somebody is doing work, somebody wanted to produce a genetic resistant variety in garden pea. So for that one, they have to use computational biology to identify the genes that they want. For example, they want to drop stress tolerance microRNA. So they have to use the computational biology techniques. So the chance computation when they have identify the microRNA using computational biology studies, so there are chances that it will get successful or it will not. There are 50% probability. So now the study should be conducted in such a way that the sequence information of microRNA should be provided so that the people who are working get a okay, fine. So objective of the study is to identify and characterize, conserve and novel microRNA and the targets in garden pea by deep sequencing. So the second is about the pathogen, to enforce a novel approach to identify, conserve miRNA and the targets in biotrophic pathogen. The third is to detect the conserve and novel mRNA in ERCP infected leaves and to identify the differential expression mRNA. So this is a 20-day-old leaf plant that was infected with ERCP pathogen. So the occurrence of symptoms after 30 days, the white fuzzy patina was observed, and this was sequenced. And during the sequencing process, they attached with RNA 3' adapter and 5' adapter during the sequencing.
I want to ask you before you go into this one. Your hypothesis is that the pea plant is subjected to the fungal infection, and therefore it compromises its productivity. That's what the observation that you have made. And in order to go after that thing, you are trying to look whether microRNAs are differentially regulated following infection with the fungal. Is that correct?
No, sir. My hypothesis would be, which I have not mentioned, the presence of microRNA in garden pea leaves. There is it would be, because here it is not applied. I wanted to identify the microRNA in garden pea leaves. That is the first one.
Why do you really wanted to identify the microRNAs? First tell me that there are so many things, right? You can identify RNAs, you can identify the proteins and all that, but there must be some fundamental logic. Why do you think that the microRNA, profiling microRNA will give you some added advantage that you don't have?
The function of microRNA is to regulate the transcription.
Yeah, but you don't have any data to propose that. Unless you have some transcriptome or proteome, that there is some something is with and without infection with the fungal, then you may say that maybe whether it is because of induction of microRNAs, they are being down regulator or upregulated. So
have some target.
Some target actually.
No, the thing is, sir.
No,
yeah, your point of view I am understanding. The first thing is that we have worked. In literature, people have worked extensively on transcriptome. The second thing is, the next step is, we have to work on microRNA because we have to develop the genetic resistance variety. This genetic resistance, for developing the genetic resistant variety, the first thing which we need is the information of microRNA. So
Wait. You don't go jump around, too much. Unless you have an evidence that because of the microRNAs, such genes are being, down-regulated, and those genes are important for, microbial resistance or pathogenic resistance and all that. So by profiling microRNAs must be compounded or supported by some kind of experimental, a background.
Sir,
not simply go to the, profiling and then I wanted to know what microRNAs are there.
no, sir. Here I have cited literature. Campo et al. He reported that.
Another thing is, I don't know what that literature suggests. All you need to say to the public and audience here is that why you have become, forced or thought about pursuing this microRNA thing, and by doing so, what is the outcome that you would expect?
Sir, the thing is, I wanted to provide information to the people who are working on garden pea plant, on microRNA. Since this information is not a sequence information I am talking about. If, for example, some person from any labs wanted to develop disease-resistant plant, then how could he get the information about the microRNA sequence?
That is not a scientific question you are asking in that case.
For that you have published a paper, information is provided. This is a thesis defense.
Yes, sir. So
synthesis of information properly. So just your thesis cannot be just based on some information.
And that information cannot be a resource for others to pursue. So you need to have a kind of, a hypothetical driven, experimentation such that you could be able to, hypothesize something, and here is what I got, and here is, this is something take home message.
So one thing I would like to mention that about that. Every lab sent their data. Every lab sent their samples for sequencing. You will get the data. The sample gets sequenced, and you will be getting the information. So after getting the information, how will you make sure that the information that you are got from that one, for example, next generation sequencing information on microRNA is reliable? How will you make sure?
Yeah, but that is out of
no, sir. I am coming to that point. I am coming to that point. So how will you make sure? Because human do mistakes. Something they will replace the samples. Then the data will come to you. You don't know what the data is because they won't give you any script. They don't give any. They will show you that this pipeline is followed, but they don't tell you what steps they have taken. So you publish those data based on your data. The other people invest their time. So human errors happen. So sir,
No. You have wrongly answered. Say you asked a question and you wrongly answered actually.
Sir,
So how do we validate it? We validate it by doing another set of experiments in the same lab.
I am not talking about the lab, sir. I am talking about next generation sequencing company.
Okay, so we got some data from NGS company
Yes,
in our lab.
yes, sir.
It is our duty to first validate that lab. Let's say if you are working on transcriptome, you will validate your data with real-time PCR for at least few genes. If you are working on microRNA genes, you will do real-time PCR for a few selected microRNAs, whether whatever you are showing in your data is actually validated by at least two techniques.
So here the problem over here is if their species are related, like garden pea and glycine max. So you will be having a conserved sequence and you will be getting the same information.
No. You will not have even the same thing with two different varieties, tolerant versus sensitive varieties. Forget the two different species.
No, species that is closely related. When you are designing a primer, you will be designing using a conserved sequence, right?
No,
so when the
no.
You don't mislead the people that NGS data will be anything which really have good reliability, low reliability. The data comes with precise information. What is the reliability? All the vendors they give you. So it's not that they give you anything and you are at loss what to make out of it. The raw data even has a quality data. You must be aware. If you have analyzed the data, please tell the audience that even the raw data has a quality aspect. You can choose the good quality, bad quality. It's the user.
so here I said that human error. I have a specified human error. Human error might happen, sir. I am not targeting about the human error might happen. So because of that one, we will be getting the data. So there is an essential need that someone should be there who can check the data. So normally when they are analyzing the data, when you are sending the data to the sequencing company, they have pipelines, right? So in that pipeline, they will keep your data. Sir, let me finish that. So sir, the problem over here is there is an essential need that someone should find out a solution wherein you will be placing all the script and all the procedure to the audience, so that when somebody is getting having a microRNA, they will take those script and this all the information which is available and carry out their work in their lab, so that at the same time they are having reliability.
Nobody's questioning on that one, but my question here is that your hypothesis where you started is really, like a clarity has to be given. Why do you wanted to go after microRNAs? That you have to, make a strong case so that you can go further. In that bioinformatics, we are not even there yet.
No, microRNA information is not available in the literature.
So
And
literature means what do you mean?
What
Have
is
you
it?
checked the Millbase database?
I am
Millbase
talking about
contains the all known plant and animal microRNAs experimentally validated as well as predicted. Did you check that Millbase doesn't have any microRNA from garden peas?
yes, sir. This is the first time I am telling you. No, I am the one who is doing this thing. There is a gap in the literature regarding the information of mRNA. So I am the one. That's why I am telling you. I have sent to Mirbase also.
How many samples did you sequence here?
Sir, 2 samples, sir.
Just 2 samples?
What
Yes.
are those two samples?
It is non-infected and infected garden
That is
peas.
not enough.
But sir,
How many replicants?
sir, actually,
Let him first present, let us hear from him first, then we'll go for the.
yes, sir. I did pooling of the sample.
Okay. No, you present your data. We have enough time to discuss on this.
So here the objective, as I said. So the data that has come to from miRNA, raw data is uploaded to NCBI, and this is the NCBI SRA.
Now, where is your
SRA.
name on that? Is it data is on your name?
Yes, sir. It's on.
It's there. Your name is written here in the CBI.
But how many reads you got? That's what we need to know.
Sir, let me complete those things. Then only you will get complete picture what I am talking about. After that, we will have a discussion.
Okay, go ahead.
The screenshot was not full. I have not taken, but it is on my name only. This ID, PRJNA, it is on my name only. First thing after coming the data, I did FASTA QC analysis, because sequencer, the QC report obtained from various sequencer will focus on the problem that arise from the sequencer itself. So FASTA QC generates the QC reports that identify problem in our static library. And then I have utilized ASIRNA workbench. It is an application. It is a Java-based application. With that application, I have removed the 5' and 3' adapter from the sequence. So the million. Then after that one, I have utilized RFAM database to download the non-coding RNA such as rRNA.